today we talk about, or the remainder of the lecture, we talk about the prediction of the effect of mutations, and the effect of mutations to be seen against the background of, I showed you this one, number of residues aligned, percentage sequence identity. If I have 100 residues aligned, 67 can change and I maintain structure. Now, what I want to talk about essentially is 1 has an impact. Here I say 67 don't have an impact. So essentially this is, we know not all 67 don't have an impact, right? Let's go back to this issue of what changes are on the level of the genome. I'm sorry that things shifted a little bit here. The types of variants you have is a single change here. Substitutions, you have insertions, you have deletions. Sorry, this changed completely the slide. Then you have duplications. Here you have the C, a double. Or you have inversions where you essentially pick the complement of the nucleotide. That's on the level of the gene. On the level of the protein, you begin by synonymous, so those that do not change the amino acids. So nucleotide changes that do not change the amino acid sequence. And those that do change, typically called non-synonymous. Then there are nonsense, that's a premature stop, for instance, so they're not creating the same protein sequence. Insertions, deletions, duplications is the same as on the DNA level. And then there are inversions, they don't make sense on the protein level, it's just a different amino acid that is taken. But ultimately this is sort of the same thing. A few acronyms. SNF is now what people use to describe single nucleotide variant. So one nucleotide change. Historically, people use SNP, single nucleotide polymorphisms. Today, they are often used interchangeably, as I say. Historically, the SNP had an attribution to populations. So SNPs were things that were variants that existed to at least 1% of the population. The reason why that was essentially is simply sequencing methods had mistakes. So everything below that you simply said were an error. That's why SNP was sort of invented this way. But today people really don't use that anymore. They still use the term SNP. Some people use the term SNP, but ultimately they talk about SNFs. SAFs is now what people use, or most people use, for single amino acid variants. So these are the non-synonymous variants, the non-synonymous SNFs, or just as mutations, they could also be not reachable by a SNF. I get to that in a moment. And then there's this language of 19 non-native. This means there's the native amino acids and all 19 amino acids that are not the one that you observe in the native protein. SNF possible ultimately is simply the question, can I get to an amino acid change with one nucleotide change? Yes or no? Not. So not all amino acid changes, not all 19 non-native amino acids you can reach by one SNF. Okay? Some you can, some you cannot. The ones you can essentially are the ones that are called SNF. We know some examples, shown here is the hemoglobin example, where a single amino acid change leads to a dramatic effect. In this particular case, the dramatic effect affects the cell, affects the organism. The carrier of that variant, a single amino acid variant, is going to be sick. Okay, so the entire system is affected. That is affected by simply the aggregation. That's the normal hemoglobin, and that's the aggregated sickle cell. Sickle cell anemia is the disease, and that comes off a single amino acid, a single SAF. How can synonymous SNFs have an effect? So those SNFs that in fact do not change the amino acid sequence, how can they still have an effect? There's a variety of mechanisms, yeah?
Maybe translation efficiency? Because
yes, that's one. So the way things are expressed are changed simply through changing the signal, the way it's done. And ultimately another one is sort of copy number variants that we see a lot of. Yeah?
And maybe some binding site obstruction? Maybe there's some on the DNA level binding component that is not reachable anymore? Which
is sort of the same thing, right? So you essentially
binding to DNA.
Yes. So essentially you change the way the protein is expressed in the way, or essentially specifically done the copy number. So there's a variety of effects you can have. But there are more examples than that. So now the next issue is, what is a variant effect? So you have a sequence variant, and from now on, I'm going to talk about SNVs. So I'm going to talk about changes of a single amino acid. How can you classify whether what the effect is?
Well, ideally you know exactly what the organism is like before, with exactly the same DNA, and then...
And on your issue, Paul, you already have an important point here. You say organism, right? So we're talking about proteins. Human, we carry 20,000 proteins. So one way would be to say this sequence variant, the single amino acid, affects me in a visible form, right? Another way would be to simply say it affects some of my proteins, right? It could be that one particular cell has a problem and that you don't see that in me. It could be that, yeah?
If it's synthesized at all or produced at all?
So
when you observe a mutation on the DNA, can you still capture the protein by means of, sequencing RNA?
No, but so again, yes, you're right, but somehow we assume, I'm not entirely sure this is at this point, so 100% we have not proven it because we still don't have an entire directory of all the proteins in human. We are saying this is 20,000, but nobody has a finger on all 20,000 at this point, an experimental finger. But I assume that every single one of these 20,000 is expressed in some cell. So if we had an effect in some cell, you would somehow see it.
If we know in one condition we have this protein produced and we induce a mutation and it's no longer produced, I would assume there's some lethality to this mutation.
Whatever is lethality. So the point is the word lethality. Lethality gets you into this thinking of Paul,
yeah,
where you look at the organism, in effect on a visible difference of the organism.
Will it fold?
Will fold is yet, by the way, is yet a different story, right? So it may be that just, I can't remember whether it was you or somebody else who talked about binding site. So you may affect the binding, so you may fold very well, and by just affecting the binding site, you would sort of affect the way you bind. No, you don't have a binding site, no matter whether you should look nice if you don't bind, right? And so there's a variety of these effects. There's molecular function, structural stability, and this is ultimately the point of it will fold. There's something like pathogenicity or disease risk. This sort of affects size, and this sort of generalizability. So there's a scale of different things that we are going to integrate over. We're going to mostly talk about molecular function in the course of what I'm talking about. I'm going to look also at other aspects over the course of the next two lectures, but our driving example, most of the samples we're going to talk about are using as a background in the data set are sort of molecular function based. The first generation methods use a very simple idea, and that simple idea goes back to this one. The first method that existed is SIFT, and before I sort of explain what it does, at Singapore now, who has an immense amount of citations from this very paper from SIFT, and SIFT essentially simply, I should have blown this up a little bit more, essentially it takes the Blossom matrix and says there's a threshold. In the Blossom matrix, if you are more likely to have to observe that variant than some threshold, you say it's neutral, this variant, and it's less likely in the Blossom matrix, you say it has an effect. Okay? Yeah?
Two things. First of all, it is based on what occurs.
Yes, absolutely.
Because that's how the Blossom matrix came up.
That is correct.
And second of all, is there, or is in the paper some evidence that the threshold actually holds true?
Yes, so in that particular paper they have, I can't quite remember, but less than a handful of proteins for which they have variants. So there's a relatively small set of variants. They choose one particular parameter, one, and they simply show that it looks right.
Because in order to prove it, I would have to,
You need some. Totally right. And in that paper they have some examples, and they verify that. So they argue for the threshold without those samples, and then they simply say, again, you optimize one threshold, is totally fine for these. So this is a completely clear story. And that gets us to the next paper. And it goes back to Shamil Srinyaev, who did an excellent job, in fact, integrating the specific family alignment information in two ways. The first one was to compute an index, PSI, he called it, that sort of essentially a very, very fancy conservation rate, a very fancy conservation rate. Once you have this fancy conservation rate, you can simply say, is it conserved, yes or no, in that particular family. So that's beyond. It is still a single parameter, but it's beyond the idea of a blossom matrix because it's specific to that family. Okay, you have to do an alignment first, but for the blossom matrix you don't. So the beauty of SIFT is very fast. You just look up these numbers for the 20 amino acids you have, it's immediate. Well, here you have to run an alignment method, and a blast on today's database is an hour of CPU. So this is absolutely not instantaneous, yes?
Well, then, do you get one sequence and then finds the family itself through BLAST?
So the BLAST essentially is, you're not looking at the variant. Yeah. So you're looking at take the query and you align the query against the database, and then you look up, is that particular variant that I have compatible with what I see in that family at that position?
With that family meaning?
From the original sequence, from the native sequence, from the non-varied sequence. It may be a good point. It may, but actually the way alignment methods work, that now we get back to my initial statement, one SAF, one amino acid difference will not give a different, in the vast majority of cases, the alignment will be the same. If you have a sort of peptide of 5, this is a very different answer. But for a typical protein of a few hundred residues, that does not change the alignment or does not change what it finds. So the PSI-BLAST profile will not differ really. Will be differ a little bit, and you could use that possibly. But this is not the idea here. The idea of SNAP-S3D, published by John Moult's group, was to, if for my protein I have the 3D structure and I sort of do it differently whether I'm exposed or inside, then that gives me additional information, that gives me slightly different thresholds, still a simple thing. I still do not need many parameters, I still don't need any large data set. I can sort of still establish in principle for a small data set that there is an effect, a difference between inside the protein, outside the protein, and then I can handle these two in a different way with two different parameters, right? This is essentially the idea behind it. They do a little bit more because they essentially compute the stability through the internal binding. And then they say, does it break that stability? But again, it's something that is fairly simple. For fairly simple, they actually use a relatively large data set already. So then we get to the next issue, yes?
But Tripolofans, so essentially we're looking at the PSSM.
Yes.
Okay.
But in fact, the way Shamil sort of computed the particular conservation, this is a PSSM with changed against PSI-BLAST by really computing conservation in a different and very meaningful way. An important different way. There's a Polyfen 2 that in fact use machine learning, and then things sort of get different. So I'm trying to get back to the origin of the way the methods worked because of a variety of reasons. So let's go into the issue of data. How do I collect data? And ultimately what I have is the same problem that we had before in the group here. We have some of changes that are on the level of the organism, and some changes that are really on the level of delta G of binding. So I have sort of minute or gradual differences between the native and the SAF. I see that the binding is slightly different. So what this illustrates here, I have a switch in the electrostatic behavior. So these are sort of largest changes again. Largest changes not on the organism, but on the level of the molecular. Now, when we look at a data set, so the first person who really aggregated a larger data set was Jana Bromberg, and she went back to a database, PMD, that is no longer maintained. And PMD, after some redundancy reduction, she found 40,000 effect and 14,000 neutral variants. So essentially giving this data set, sort of put the next slide. Imagine you have two proteins, both of which are shown to be enzymes, and both have exactly the same EC number. So they have exactly the same function. Okay? You align them and they are similar enough so that the ones where they differ, you believe, are not new binding sites, are not things that the other one does somewhere else differently, but essentially are the variants that you can allow and still have the same EC number. Then you would argue, and I admit you will make some mistakes, but then you would argue that the vast majority of these changes that I put in here, if what I really have is the enzymatic domain, are in fact residues that I can change without changing function. Because I measure the function twice, I have two experimental measurements for both. I mean, for each I have one. So I have two different independent experimental validations that A and B do the same thing, exactly the same thing. And then I can argue in the alignment where they differ, it's neutral. And you're totally right, this will not be always right. But as long as I'm really talking about the region that does the enzymatic function, in most of these cases I should be right. Most of the things I pick should be right, yes?
And don't know if I'm completely up to date with how much attention I pay, but what if it slows down the enzymatic?
That's a good point. So this ultimately gets back to the toggle and restart issue. Slows down in that sense would be a tiny change. If we in fact talk about things where just yes or no makes a difference, then we would say it's still neutral with respect to that. And if it would be a restart, I would mix it up, and you are totally right, then some of those will make a difference. Yes?
Then I have to limit my queries to the domain that's actually functional, right?
And that brings us to a lot of buts. This puts us to a lot of restrictions, and this is why in this slide what I add here is 24,000. So I'm not adding 100,000 from all the enzymes known, but since I have to play it, so I have to have so many constraints that ultimately what I add is not that much. Mind you, these are the numbers not for human. These are the numbers for everything we had in 2007, right? And still, so the gain was relatively small. And that's exactly because we had so many restrictions. So these are simply the numbers. There's effects, there's neutral, there's the ratio. We see that the ratio is different between buried and exposed residues. So buried is 0.75 and this is 1.6, very different in the same data set. And this again is something, so we are unclear whether we expect that, but we tend to argue that more changes on the surface are neutral, because on the surface you're simply less constrained. Inside you're more constrained, any change inside will affect, you have more of a surrounding inside that can change than on the surface. Yes, all the binding is done on the surface, but most residues on the surface don't bind. So this is sort of, in some sense, these numbers in that sense are not surprising. Now, what I would call the second generation method can now start. These are methods that essentially need enough parameters. 80,000, 40,000, 40,000 is not a huge data set. In particular, and Ron, you said that right, in particular when you think about the possible number of variants we have in 6,000 proteins, right? When we look at 20,000 human proteins and say on average the protein is 500 residues long, then this is 10 million possible changes, or 10 million residues in all human proteins. Everyone can change, it's 19 different positions, so we have about 200 million possible changes. And the 40,000 or 80,000 is a tiny number compared to these 200 million in human alone, and this would be all proteins, right? Okay, but at least we can do machine learning methods. By the way, the third generation method is sort of larger data sets, and that is sort of we are beginning to get into that framework. Yes?
If you look at the data set with domain information, right? You get your EC number and you parse out the domains, and you say because there is some variance between two regions within the domain, I assume those are not as rigid or don't have an effect because
exactly they're neutral.
This is restrained to domains, right? When
yes.
in this domain is most likely to be in the surface.
Yes.
If the same change
There-
within the protein buried, the change might be indeed better.
In fact, you put your hand on something that is one of the reasons why I took much more time for this talk for these talks in the past than I do now. The field has come, in some sense, to an impasse because we have this huge possible data set, human alone 200 million, and these tiny experimental data sets. Although, if we increase the experimental data sets by a factor of 10, it's still small compared to what we want. And that leads to a situation where we have now 200 methods that are all optimized on a tiny subset of what reality has. No matter what we do experimentally, we're still very, very far away from where we want to go. And the more we get into the deep learning, the worse the situation comes, because the methods over-optimize even more this tiny set of data we have. And we don't actually know what to do about it. But this is somehow why the field has come into a corner. I will maybe briefly talk about that more on Thursday. But what we see is that when we compare the methods based on the data that we have, the experimental data, they look very similar. When we look at the methods for all the 200 million, they actually look very different. But the difference between those, we don't know which method is right. Because by definition, we have the data only, this small data set, right? For all the others where we can predict for variance that we observe in human, we can predict the effect, but actually is it right or wrong? We don't quite know. What we see is that methods behave very differently on the tiny set for which we know the answers than on the sets for which we don't know the answers. Don't know what is right here, but we begin to see that something is wrong. And we don't quite know how, and that makes my talks a little bit shorter, because the field, this is very important to do, but we don't quite know how to do it. And we have over the last, say, 3 to 4 years come closer to the wall. The wall is getting heavier and heavier, and we are sort of hitting again, I believe, is what it actually is. So the person who collected the data set and started the first machine learning is Jana Bromberg. But I'd like to quickly run through a bunch of other ideas. Again, historically I've done that in an environment where there were 20 methods out there, and I could just run through 20 methods. And I'm going to run through 10 of them, but we have 200 now. So I'm further away, so I'm showing you some ideas, and they sort of reflect essentially more the beginning of things. So the first one here, SDM, comes from Blundel's lab, and that essentially is in the thermodynamic cycle of binding. You look at the difference that happens through the effect of sequence variance. There is a mutant that has a sort of delta G on binding, and Blundel's lab has done a lot to measure these the binding cycles, the binding of proteins to the substrate, and the effect of variance to that binding, and developed a method that essentially on different levels, through machine learning and through fitting, and here is more fitting of the understanding of functions or our understanding of potentials of binding, explains which mutant has an effect. Ultimately by fitting some data to some function. Pop music from the lab of Marianne Roman. Actually, there's a bunch of those that are funny from interesting names from her lab. This is just one of them. Does something like that. Looks at the same delta G of binding, but sort of tries a better or more. They were the first to sort of do this with machine learning, and then we get into iMutant from Emilio Capriotti, Piero Farazelli, and Rita Casadillo. Essentially, they begin with the same experimental data, fitted with machine learning in a way that is different from this one here, because they look at delta G, they look at the data in a slightly different way. Automute, Josef Weissman, is a way to look more of the energy of binding and look at differences in the energy of binding. So look again more at the understanding. Probian is an extension essentially of SIFT, and it introduces a concept that I will explain in a few more slides, where we look at family specific. I'm going to skip over that. here's the mutation taster. In some sense, this is Paul's idea. We look at variants within, for instance, mammals, or within human, or within some subspecies. So they looked at mammals and they said, what we see when the variant is observed in another mammal, higher mammals, at some point ape, at some point mammal, then we will say this is accepted. If not, then we will say it's an effect. There is evolutionary action from the Lichtarge lab. So this ultimately compiles potential coming out of tree formations, and goes back to something that I hope I have in a few slides from now. So ultimately the question is the variant one that I see when one major part of a tree switches to the next? So is it a change that is sort of within a subtree, or is it a change that sort of joins different parts of the tree? And they compute a sort of energy related value and use that. CAD is one of the best methods, and this is, I'm missing the quote here. It goes back to Martin Kirchner, and there's a new version of CAD now. CAD essentially says that if I sort of see it in alignments of things, then it's okay. If it is different from the set of others, from neutral variants, I simulate and I do machine learning to separate these two distributions. That's one of the best methods out there. Mutation taster is the idea that I meant. It comes originally from Boris Reva and Christian Sander, and goes back to what Provian says. So here in the slide you see that there is 4 times E and 3 times N. Okay? Now what we see in, for instance, the machine learning device in which we would use a profile as input, or in Polyphen, this situation would essentially produce the same input unit as this situation. Right? Same count. But it is different, because if you belong to the top set of proteins, you actually need an E, and that's the distinguishing feature of these proteins. If you have an N, that does not mean that N or E could be observed. It depends on who you are. Are you part of this subset, or are you part of that subset? If your sequence falls into that subset, E is not tolerated. Okay? That's the idea of mutation taster. So something that is sort of subfamily specific, a value, if it breaks your sequence, because your sequence now in the alignment, it goes back to an idea you had before, Ron. But if you align your sequence, yes, overall you will get the same set of proteins you align to, but you may belong to that part or that part. And that ultimately is the question. If you belong to that part and you have an N, then something goes wrong. Okay? That is the argument behind mutation taster. Now, there is the next idea that goes back to Debbie Marks, Chrysander. It was implemented by Thomas Hopf. And assume you have 2 residues, such as these, for which you look at the alignments. If they are close in space, then in this variance that you see here and here, you will have a reflection of the fact that they're close in space. We use that to predict contacts. Konstantin was briefly talking about that. Now, if we inverse this problem, we sort of can use that to predict contacts. So essentially we can compute the evolutionary couplings, and we can use these evolutionary couplings to predict the effect upon function of a variant. Of a SAF, not only can that thing predict the effect of SAFs, but also pairs of SAFs. So it can compute for any number of combinations of possible amino acid variants, whether they are correlated or not. And this is what is, in fact, it's the only method that can do it. I should put the new, has been published since a while. So, yeah?
But isn't a method like this like hyper-focused on super important parts of a protein, like parts that interact with other parts of the protein, or that are for binding sites?
That is absolutely true, Paul.
So if you have somewhere in the background,
that is true what you
change. I feel like you wouldn't be able to assess well enough if this changed some random amino acid on the surface.
That is true. That is true what you say. They would be, well, actually, let's just pick a random point on the surface. So let's call this random point on the surface and binding site. Then you would say, well, these are the exceptional cases where it's special. Okay, so if it is not one of the special sites, a random point on the surface that is not special, it's random. And you would argue, mostly that's neutral.
Yeah. So, but this would not...
It does, yes it would, because it sort of groups things into two.
I also would say if this is
You don't see the signal, you call it one of those random points. You see a signal, you call it one of those important sites.
And for the important ones, we have our data where we know if we can allow
Exactly.
mutations or not.
So the major problem here, in fact, is that we, in order to do this, the evolutionary coupling need large alignments. So this is essentially technically a problem. It's not easy to do. We cannot do that for all human proteins. Or we cannot easily, so the performance, so they show that this method performs much better than anything that is available, even for single SAFs, but that is valid only for the set of proteins for which we have big alignments. So the smaller the family, the more this goes wrong. Incidentally, there's a fantastic Cell publication from December from Chrysander's lab, in which they essentially do in silico, or not, in artificial variation. So they create evolutionary samples. So they create their own evolution in a tube to create samples for the prediction. And they essentially say, if it helps in our, so if we create enough samples, we can improve our prediction, and we can predict essentially structure without doing a crystal structure, by just doing mutants. And that's a totally cool Cell paper. And it seems to work. Anyway, so that brings us back to the question, how do we compare methods? And then you may say, well, we simply show the method, we show a variety of criteria for being right, and then you get a table like that. And technically that table is right. What is the problem again is what I said before. We have a tiny data set that reflects the entire universe of things, and this is a too small subset. But the other thing that is wrong with it, now I give you 1 minute, show me the one that is best. And you cannot. And in fact, the story gets even worse if you sort of have longer ones here. So if you look at more measures of performance, and you need to look at more, this doesn't capture everything. Because very often some methods are good from under one aspect and not under the other. So the story gets really complicated. And I'm not sort of taking that complication away. I don't have a solution. I'm just going quickly through one particular effect prediction through machine learning solution, where the idea is we do exactly the same as before. We have this sort of 13 consecutive amino acids. Each one of these bubbles here is sort of 21 units for 20 amino acids plus a spacer, and we have effect on neutral predicted. And the input features that we have reflect different aspects. We have biophysical features, alignment profiles, probability of residue triplets. We have, so essentially that goes back to the genome issue. We have solving stability, secondary structure, residue flexibility, and some annotation features. Then we predict, and essentially we do a binary prediction, and we look at the overall performance. For a set, you asked me what was done for SIFT, so they had these 3 proteins. For this set, we get a sort of certain performance. This is Q2, so what fraction of the ones are correctly predicted? This is precision versus recall. This is the corresponding thing for the neutrals. This is effect, so really precision versus recall. This is called something else. I always forget these numbers, these words. Interesting, what you see is that exposed is performing worse for effect than buried, and for the neutral it's just the opposite. So it's a zero-sum game somehow. You do better for one on one end or the other. You have difference, so both of these are predicted as effect. This is stronger than that. One is stronger than 2, and this strength is used to predict the reliability index. When the reliability index, you see that the higher the reliability index, the higher the cumulative performance of that number. So that is important. Then you see that, in fact, if you classify the types of effects you have into very strong ones and intermediately strong ones and sort of more neutral ones. That is the scale. On that side of the scale it means strong effect predicted. On that side of the scale strongly predicted to be neutral. And you see that stronger predictions, so more on that side, is also more red. So more severe effects are more on that side of the example, of the spectrum. Now we can, when we look at, I will present that at some later point. So then we get to some point in this work where you can sort of publish that your performance is. But one way to answer that is you look at other scores again, right? This is clearly one answer, and then you look at many more details and you convince yourself that looking at many more details, you essentially see more to it. But another way of looking at it is, I already said that most methods on the set of proteins for which we know the answer essentially give similar results. So why not look at a subset of SAFs for which the methods give different results? So I'm going to show in the next slide, I'm going to show you all SAFs, and on the next one I'm going to show you only the subset for which at least two methods disagree. Okay? And then we get this one.
No.
So again, we're throwing out now most SAFs with experimental annotations. We're throwing out every SAF. We're keeping only the ones for which two of the three methods here disagree. And for that subset, these two simple methods here are worse than random. You may ask yourself, how can you be worse than random? Ultimately by throwing away everything that works, right? You take everything where the methods work, you take it out, and then you say, bah, you're worse than random. It's totally unfair. And it is in fact true if you sort of compare these two slides. Here, SNAP sort of goes from 80 to somehow 60, right? So it pays a lot for this. So these are much tougher cases, and SNAP performs much worse. But now we have a set where, in fact, for the ones where the answer is not clear, already given by simple methods, SNAP does a much better job, because it's the only one that is better than random. So this is a simple way of defining your investment in terms of time was worth it. Max went on, sort of put in new features, put in new data, used also coevolution signal, and many, many, many more. Got to a new version of SNAP, SNAP 2, that performs better and has a variety of advantages. For instance, you can also use it if you don't have alignments. This is sort of the idea here, much less accurate, but you can actually essentially predict the effect of a SAF from more sequence-based features too. That gets us